Let's begin with a little refresher of anatomy. And so there are three parts of the ear. We have the external ear, the middle ear, and the inner ear. The external ear consists of a pinna or the auricle, and it has the external auditory meatus or canal and the tympanum or the tympanic membrane. This portion, we have the transmission of airborne sound waves into a fluid-filled inner ear, and it helps to amplify sound energy. The middle ear transmits that airborne sound waves into the fluid-filled inner ear, and it also helps to amplify sound energy. And then lastly, the inner ear has two different sensory systems. We have the cochlea and the vestibular apparatus. The cochlea contains receptors that are important for conversion of sound waves into nerve impulses, and this is what is responsible for hearing, whereas the vestibular apparatus is necessary for the sense of equilibrium. So if we look at here in this image, so this is divided into the different portions. So here we have the external ear, the middle ear, and then the inner ear. And where the middle ear, we have those auditory ossicles that are going to start the transmission of mechanical vibration that will travel into our inner ear. So the sound waves are going to be funneled by the pinna or the auricle into the external auditory meatus, which channels them to the eardrum or the tympanic membrane. So the middle ear has the air-filled cavity that's between the tympanic membrane and then of the cochlea, and has the three different ossicles of the malleus, incus, and stapes, where vibrations are going to be transmitted along these bones. So the stapes, which is right here, is going to be attached to the oval window that transfers vibrations into the cochlea. And then the muscle that's attached to it is going to help to dampen the stapes if the sound is too intense. So such as if you go to like some type of concert, that muscle helps to not have that sound be so intense when it enters our inner ear. For the neural aspects of hearing, what we have are two different localizations. So the first is where is the sound coming from, and then the identification of what is that sound. And so this has to do a lot with memory and with emotional state as well, and helps to recognize familiar voices or sounds that we've been exposed to throughout our lives. Sound waves are going to travel through vibrations of air, and so this will consist of compression and refraction of air molecules, which we'll get into now. And so sound waves are characterized by frequency, and so this is measured in hertz. High frequencies have higher pitches, and so for humans, we range from 20 to 20,000 hertz. And intensity or loudness, this is measured in decibels, where it has to do with the amplitude of the sound wave. Optimal range for us is going to be between 0 to 80 decibels. So both frequency and for intensity, though, these will start to decrease as we age due to exposure to loud sounds throughout our lives. Here, let's look at properties of sound waves. So first, let's begin with the pitch, which depends on the frequency. So if we have the same loudness but a slower frequency, that's going to be detected as a low note to us. Whereas if we have a higher frequency, this is perceived as a high note. Now, intensity or loudness depends on the amplitude. So a small amplitude in sound denotes a soft sound, whereas a higher amplitude is going to detect then a louder sound. And then one other quality that we have is timbre, or number of overtones. So if we listen to a pure tone, this is going to come out as a smooth sound to it. However, if we have different overtones that are laying on top of each other, this can interfere with one another so that we perceive kind of like a muffled or a jarred sound. This table denotes an idea of what common sounds are in terms of their loudness on the decibel scale. So soft sounds tend to have lower decibels, so like rustling of leaves, which would be considered to be at 10 decibels, are going and then compare that to, say, normal conversation, which is at 60 decibels, is about 1,000,000 times louder than that of nothing. And then takeoff of a jet plane, this is at 150 decibels, where this is 1,000,000,000 times louder when we look at those differences in sound waves and loudness. So airborne sounds are going to be transmitted from the tympanic membrane vibrating when it's struck by different sound waves. So then the middle ear is going to transfer that vibration through the ossicles, again, of the malleus, incus, and stapes to that oval window within the cochlea of the inner ear. The waves in the cochlea then are going to set the basilar membrane in motion, where there are hair cells that are going to bend based on the frequency of which they are activated. And so these are either going to deflect up and down, and so start with the perception of sound depending on what frequency they are being stimulated at. Also, mechanical deformations occur at the hair cells that are transduced into neural signals that then transport into the temporal lobe for the auditory cortex, where we have the perception of sound. So as sound waves travel through the external ear, they're going to start vibrating that tympanic membrane, which then will start vibrating these auditory ossicles and then start entering into the cochlea, which is what's represented here. All right, so there are 3 different chambers that are found within the cochlea. The upper chamber here is the scala vestibuli, and this is part of the bony labyrinth. And then this is the lower bony labyrinth of the scala tympani. So both chambers are going to be filled with a special fluid called perilymph. And the cochlea also has a portion of the membranous labyrinth called the scala media, which we find here. And this is also considered the cochlear duct, and this is filled with a fluid of endolymph. So the perilymph is found with the scala vestibuli and the tympani, whereas the scala media, the middle layer, has the endolymph. And so they'll have different concentrations of ions. And so that's what will be responsible then for vibrations and movement of those hair cells. Within the scala media, there are 2 different membranes. We have the tectorial membrane, and then we also have the basilar membrane. So the tectorial membrane is where we will have movement of the hair cells with these fine little proteins of stereocilia. The basilar membrane is where we'll have their axons that are going to project and then form eventually into the cochlear nerve. When we have activation of these hair cells then, what will happen is that potassium channels are going to open and cause depolarization, and so that's going to cause action potentials that are going to be based on different frequencies. So this image is showing that sound waves are transmitted through the cochlear duct at locations that depend on the frequency of the sound. So low frequency sounds are going to travel further down the spiral of the cochlea and are going to be interpreted here towards the apex of the cochlea. High frequency sounds are going to be interpreted within the base of the cochlea, and then middle frequency sounds will be somewhere within the middle. So the bending of hair cells on the basilar membrane is what is going to be perceived as what frequency of sound there is. So the spiral organ is made up of the basilar membrane, the inner hair cells with sensory fibers, and then that tectorial membrane. All right. So here we have the tectorial membrane, here we have the basilar membrane, and then here we have the different hair cells. The sensory hair cells are located on the basilar membrane that project into the endolymph of the cochlear duct. So the hairs are stereocilia that are large microvilli that are arranged in bundles. So when sound waves enter the scala media, that middle layer, the tectorial membrane, this right here, will start to vibrate. and that will cause bending then of the stereocilia. What happens is that calcium, so this is going to then open potassium channels that are facing the endolymph. So potassium is going to rush in and cause depolarization of the cell. Then the neurotransmitter is going to be released, and the neurotransmitter then is glutamate onto the sensory neurons. Potassium then returns to the perilymph at the base of the stereocilia, where then it will begin to recycle. The greater the amount of basilar membrane displacement or movement and bending of that stereocilia, the more glutamate that's released, and so that will produce a greater action potential. However, if it bends too far, what happens is that those little stereocilia can actually break, and then that is what will begin the cause of hearing loss. And so when we have the neural pathway now going from that inner ear, then we'll have the vestibular cochlear nerve is going to synapse then onto the cochlear nuclei within the medulla oblongata and in the pons. Then this information will travel into the inferior colliculus of the midbrain, where then that information is going to travel up into the thalamus and specifically the medial geniculate body. And then that information from the thalamus is going to synapse onto the auditory cortex of the lobe. Now, one thing to note then is different regions are going to be activated within the temporal lobe based on what frequency. So low frequency sounds are going to perceive towards the lateral portion of the temporal lobe, whereas high frequency sounds are going to be perceived more medially. So for hearing impairment, these are where sound waves are not conducted from the outer to the inner ear. And so one example might be that if we have excessive buildup, as I'm drawing in here, of earwax within the external auditory meatus. One other reason might be that there is fluid that we have within the middle ear. Also, there could be damage to that tympanic membrane, or there could be overgrowth of the bone within the middle ear here. And so all of these are going to impair hearing of sound frequencies. However, these can be helped with a hearing aid. So I'm going to write plus hearing aid. And so again, these can be reversible. Over time though, we might have damage to the bones anyway, and so that's why a lot of times we see older individuals that have to wear hearing aid because of damage that's done over time. The other type of hearing impairment is sensorineural or perceptive deafness, and this is maybe due to nerve impulses that are not conducted from the cochlea to the auditory cortex. And so this may be due to damaged hair cells a lot of times from loud noises over time, and may only impair hearing of particular sound frequencies and not of others. Typically, high frequency sounds are what are affected first, more so than of low frequency sounds. And so there might be some hope for those with cochlear implants. However, a lot of times those are not as effective though for all types. So injury and loss of hair cells caused by intense noises will affect portions of the organ of Corti within the 3 rows of the outer hair cells. And so what we're looking at here is in panel A, we have a normal guinea pig, all right? And so here in figure B, what we're seeing are the guinea pig exposed to 24 hours at a decibel of 120. And so this is very similar of going to that of a rock concert. And so what you start seeing is that the hair cells have been damaged, and so once these are damaged, these are irreversible. And there is some hope for stem cells, however, not on the market yet and not 100% effective and treatable for humans yet. Equilibrium is also part of the ear, and so this is now part of the vestibular apparatus, which is found within the inner ear. And so this consists of the semicircular canals, which are going to be important for detecting rotational accelerations or decelerations in one of three directional planes. Also, we have the utricle and the saccule, which are important for detecting changes of linear movement in a direction, and they provide information of head position in relation to gravity. So we look at the vestibular apparatus, so again, part of the middle ear. However, so the cochlea, this was responsible for hearing, and this is thus where we have the auditory nerve synapsing here, but now the vestibular nerve is going to synapse then now on these semicircular canals that we have here, which are found in three different planes, and then also with the utricle and the saccule. So what equilibrium is that it's due to mechanical deformations of hair cells due to movements of fluid within the semicircular canals as well as within the utricle and saccule. This gives rise to then the vestibular nuclei in the brainstem, where we'll have the convergence of the vestibular cochlear nerve, and then also to the cerebrum for use of maintaining our balance and posture, also within controlling eye movement and perceiving motion and orientation. So the inner ear again consists of a bony labyrinth surrounded by a membranous labyrinth, and between the two is a fluid called perilymph. Within the membranous labyrinth, then we have the endolymph. And so the endolymph has an unusually high concentration of potassium, which is what is responsible for producing depolarization. So that when we have movement of the hair cells, then we'll have either an increase of potassium entering the hair cells. Once these hair cells are stimulated, the stereocilia are going to bend toward the kinocilium. And so here in this picture, so here we have the kinocilium and then the stereocilia. So these are going to be connected by these little tiny strands called tip links, and they're just small protein strands again. And so when they bend, we'll have potassium channels open, and potassium is going to rush into the cell and depolarize it. The cells then are going to release neurotransmitters that depolarize the sensory dendrites in the vestibular cochlear nerve. Typically what we see is that glutamate is the neurotransmitter that is released. So the bending away from the kinocilium though is going to hyperpolarize the sensory dendrites, and then what we have then is this code for detection of direction. So here, if something is stimulated, see we have the bending of the stereocilia as well as the kinocilium. However, when we bend in the opposite direction, then we have the inhibition, and so that's going to decrease the amount of potassium, and so it's going to actually hyperpolarize the cell. The otolithic organ helps to provide information about linear acceleration. And so this is now where the utricle and the saccule come in. And so the utricle is responsible for horizontal information, whereas the saccule is responsible for vertical information. And so these are just specialized epithelium that are called the macula, and that's what houses then these hair cells. So the stereocilia are embedded in a gelatinous otolithic membrane, and it contains many crystals of calcium carbonate called otoliths, and these are literally ear stones. And so the purpose of them is that it helps to give some density for these hair cells so that they're not constantly being activated and are hiring and firing at such high frequencies. The semicircular canals project along 3 planes to detect rotation. So each canal contains a semicircular duct that's filled with endolymph. So at the base of each duct is in a large area called the ampulla. And so the hair cells are embedded in the cristae ampullaris with a stereocilia that's cupped in a gelatinous cupula. So here we have our example of an ampulla, and so here would be the cupula where we have those stereocilia projecting. And again, so as we rotate our head, then what we have are different rotational activations of our semicircular canal. So for instance, this one, if we're turning our head to the right, is going to activate then the left horizontal semicircular canal as well as the right horizontal semicircular canal. And so these are going to be due to fluids that are moving within the semicircular canal and bending of the cupula in the hair cells. So with pathway for equilibrium, what we have is that the vestibular cochlear nerve is going to synapse in the vestibular nuclei of the medulla and then into the cerebellum. So the medulla is going to send neurons to the ocular motor area of the brainstem, and it's going to also control eye movement now down the spinal cord to help to adjust body movements in terms of the location of the head. So 2 conditions that can occur are nystagmatism and vertigo. So when a person's body is spinning, eye movements are toward the opposite direction of the spin and help to maintain a fixation point. But when the body comes to a stop, the cupula is still bent by fluid inertia and the eye movements are still affected. And so the jerky eye movements are called nystagmatism, and then nystagmatism then can cause a loss of equilibrium due to vertigo. A lot of times these are associated with bouts of dizziness, pallor, sweating, nausea, and vomiting. And it's similar to what some people experience with motion sickness, like within a boat, in a car, or on a roller coaster. It's because those the hair cells within the cupula are bent, and so the eye movements are creating it to be such that they are perceived as going in that direction.